Next, we will perform experiment 4, which investigates the environmental conditions required for seed germination. This single experiment is designed to test the importance of water, oxygen, and suitable temperature for successful germination. The objective of this experiment is to show that all three factors, water, oxygen, and warmth, are essential for a seed to germinate. For this experiment, we use 4 test tubes labeled A, B, C, and D. Each tube contains 10 cress seeds placed on cotton wool. Tube A has dry cotton wool to test the absence of water. Tube B acts as the control, with moist cotton wool and normal room temperature, providing all the necessary conditions. Tube C contains moist cotton wool covered with a layer of oil to prevent oxygen from reaching the seeds. Tube D has moist cotton wool but is placed in a fridge at around 4 degrees Celsius to test the effect of low temperature. The required materials for this experiment include 4 test tubes, a test tube holder, cress seeds, cotton wool, water, oil, and a fridge to maintain low temperatures. The procedure begins by placing the seeds on cotton wool in each tube. Tube A is left dry, Tube B is kept moist, Tube C is kept moist but covered with oil, and Tube D is kept moist and placed in a cold environment. Tubes A, B, and C are left at room temperature, while Tube D is stored in the fridge. All tubes are left undisturbed for 3 to 5 days, and the moisture in tubes B and D is maintained by adding water if necessary. After this period, the seeds are observed for germination. The observations show that seeds in tube B germinated successfully, while seeds in tubes A, C, and D did not. Tube A failed to germinate because water was absent, which is necessary to activate enzymes and start growth. Tube C did not germinate because oxygen was blocked by the oil layer, preventing respiration and energy production. Tube D failed to germinate due to the low temperature, which slowed down enzyme activity and metabolic processes. Tube B, with all the required conditions, germinated as expected. The results confirm that water, oxygen, and suitable temperature are all essential for seed germination. If any one of these factors is missing, the seeds remain dormant and fail to grow. And, from these observations, the results can be analyzed as follows. Germination requires the presence of water, oxygen, and a suitable temperature for the seed to begin growing. In tube A, water was absent, so the enzymes inside the seed could not be activated and germination did not occur. In tube C, oxygen was absent because the layer of oil prevented oxygen from dissolving in the water and reaching the seeds. In tube D, the seeds were kept at a very low temperature, which was not suitable for proper enzyme activity. In contrast, tube B provided all the necessary conditions for germination and served as the control, so the seeds germinated successfully. The conclusion of the experiment is that successful germination depends on the presence of water, oxygen, and warmth. The absence of any one factor prevents the seed from germinating, highlighting the importance of these environmental conditions for the growth of new plants. Also, note that for fair testing, it is important to control variables. The number and type of seeds should be consistent, light conditions should be controlled when testing temperature, seeds should be healthy and of similar quality, and only one variable should be changed at a time.